So thanks Erica and Bertie for inviting me to give this little data presentation. At Nisqually Reach Nature Center, Terrence Lee, Daniel Hull were involved in getting us permission to do this whole project. And Margaret Hummerding from the Nisqually tribe was always there. We've done some spot prawn work with her as well. Other people to thank or recognize. Oh, page down. Some of the species ID work was done, helped out by University of Washington people, Greg Jensen, Leslie Harris, Natural History Museum of LA County did all the polychaetes. And Kristen Holtgren from Seattle U helped with the crustaceans. NOAA divers put the arms in the water, pulled them out. There's a whole list of divers, thanks to those guys. The primary photographer was from NOAA. He was an IT guy named Mike Boehner. And the video stuff that you're going to see today was a NOAA guy too, Paul Hillman. So what I'm interested in, and I think you all are interested in these aquatic reserves, are identifying biodiversity. What are the species level biodiversity indices that you can come up with in your particular areas or in the Salish Sea. This project involves putting these gray things. Oh, can you see my little marker? Oh, amazing. Whoa. Yeah, that's an arms down there. It's a stack of plates. I'll show you better pictures. But basically, it's once you haul them up after 1 or 2 years of fishing in the sea, you look at animals that are greater than 2 millimeters, and the rest of the stuff you scrape off and you send off to the DNA lab to be identified using metagenomic techniques. The next slide shows you, this is a close-up of the arms itself. There's a quarter on top of it. You can see the quarter. In metric system, there are 230 millimeters on a side, and there's a little 12 millimeter gap between them. These were designed in Hawaii. Rusty Brainerd started a project in the tropics. In the tropics, they were at the very bottom. Oh no, go back. Oh, they were for monitoring reefs. What is the biodiversity of the small critters next to reefs or in reefs? And they were autonomous reef monitoring structures called ARMS. So a lot of hot air all about that. So anyway, that's the ARMS. So now I want to show you, this is sort of a summary of the ARMS project. It was photographed, videotaped in Nisqually, and I think it could be very useful. Biodiversity is what holds an ecosystem together. Gives it resilience during change. This is a project we're trying to catalog biodiversity in Puget Sound. We're trying to look at the small animals that are overlooked and understudied in Puget Sound. So about 2.5 years ago, scuba divers went in and put in these structures called ARMS. They're actually a series of plates, plastic plates with openings between them. Things came and went over 2.5 years. Things attached, things swam through, like little invertebrate hotels. So today we brought them up. 2 divers went down. They dropped a cage on top of the arms, invert it and bring it up. So we've captured what was living in there. They're carried in totes, so everything's still alive. So when we bring them into the laboratory, we unbolt the thing, we take the structures apart, so all 9 plates are swished, and we get all mobile animals into a big tote. We sieve them out. Anything bigger than 2 millimeters is taken into the lab. We get to pick through and try to identify what's been down there. There's crabs, there's shrimp, we see sea stars, sea urchins. We saw 3 flatworms that just ooze their way around on the plates. One of the coolest inhabitants of this thing that we've seen at every location is this pygmy Eula shrimp. It's a 1/2 inch at most. And we found females are just totally full of eggs. And we got 150 of them on the spot. Yet, who knows about that? I didn't know about that. I'm excited to be able to photograph those, put them in the category. So it's a 2-step process. We identify all the species we can by eye that are greater than 2 millimeters. Everything else, then, we extract DNA from the unknown things that we can't quite see. So by gathering information here, by doing the first inventory at this level in Puget Sound, it lays the foundation for future work. What is there now? What is there in 10 years? And I think by keeping an eye on this inventory, we're better able to look at models of
ecosystem change, and how is the ecosystem responding at all the different levels?
There. Now what do I do? Stop share. There. Did you hear it and see it? Yeah. All right. So we'll go back to the PowerPoint. Any comments? I'm going to cover pretty much the whole process briefly, but in the end, if there's more questions about the process, let me know. So I'll go back and I share PowerPoint.
That was super, Gary. Really appreciate it. That was really cool.
Thank you. That was a lot of fun. Paul Hillen is an amazing guy. I guess this is what I'm sharing. How do I get to my... oh no, I'm doing PowerPoint. There. Now, can you see that again? How we done? And you can hear me? Amazing. Sorry, you have to listen to me. Anyway, so that's an arms. Back in 2014, they cost about $300 to have these precisely cut, and all the hardware had to be stainless steel or plastic, just because of salt water. I have a funny little thing on my screen. Do you see this green square?
Yeah, Gary, I think we're seeing like a zoomed in. I think can you drag the square over the whole PowerPoint? Can you drag the corner over? Because we're seeing just a corner of the PowerPoint screen.
Oh, you're right. Oh. Weird. So
yeah, I think that green square shows you what we're able to see. There you go.
Oh,
perfect.
There. Okay. So as I mentioned in the thing, so now you see the whole slide maybe, right? Okay. So this was developed by Rusty Brainerd, a biologist in the Hawaiian lab. And again, he emphasized looking what you normally don't see, and therefore it's understudied. And yet they have these cryptic organisms have an important role in ecological systems that we're trying to study. So that was YouTube. I'm not even going to try another video. Forget that one. That was the best one. So in 2015, we put 3 arms. They go in about 30 to 40 feet down, and they're 10 meters apart. 3 of them at Shannon Point by Western Washington's Marine Lab, out at Nia Bay in an enclosed bay, at a bay across in Manchester. I don't know if y'all know where that is, where the ferry goes by from Bainbridge or Bremerton, and then down, of course, in your neighborhood. They soaked for, I said fish before, they soaked for maybe 1.5 to 2 years, and then again we drop a net on them and flip them over and try and keep everything alive and intact in the container. There's big habitat difference between all 4 places. In the north at Shannon Point, that was a high energy place, lots of sand and cobble. We lost 2 of them. We only retrieved 1. Manchester, and then all the other places, Manchester, Nisqually, Nia Bay, we could retrieve 3 arms at each place, but they're all different. Manchester is sort of a semi-protected area with just moderate current. Nisqually was a protected bay off of Anderson Island. Low energy, a little muddy sand, and Nia Bay was a protected inlet, and there they had moderate energy with high energy storm stuff coming through there. So that plays in what characters we're going to see, what animals. And this is the Nisqually Point, or my glasses. So that's Anderson Island, is that right? And that's East Oro Bay is where our dot was, where our 3 arms went in. So just jump to the results. Here we go. So Shannon Point, only 1 arms, but we saw 42 species and 511 animals. And that's pretty remarkable because going south at Manchester, we only saw 21 species on 3 arms. Again, Shannon Point, we only found 1. The rest were swept away. Over 500 animals at Manchester. Nisqually, we saw 29 species. and over 600 animals. So Manchester, Nisqually, about the same number of animals on 3 plates. And actually, I guess about the same number of animals at Nia Bay. But we saw many more species at Nia Bay. And we can talk about that sometime. At Nisqually, again, we saw 29 animals, 611. And this is sort of a little list of all the animals we saw. We'll be going through some of these briefly, but the species we saw the most of was this pygmy Euless shrimp, almost 250 animals. And they're just teeny little things. They're very cool. We saw jingle shells, Pododesmus or something. I have a bigger, yeah, I'll verify that. And then the third most likely, or it was the dock shrimp, Pandalus dani. This is the little pygmy Eulid shrimp. Again, taken in the bottom right corner, there's a red marker that says 10 millimeters. So that's the length. These animals are maybe 12 or 15 millimeters long. I have some good shots of these. Again, they're fantastic coloration and teeny. And the first time we saw them, I say in the video, clowning around, they were, I couldn't believe it. I thought these were all juvenile shrimp of some time before I talked to Greg Jensen. And then in the same day, oh my gosh, here's one and it's full of eggs. So that was the adult size. I learned something. And this is the jingle shell. Wait a minute, where's the genus? No, I don't have it. I think it's Pododesmus. Anyway, jingle shells, they're actually a bivalve. There's 2 shells, but the second one is very reduced and they attached. Adults get to be about 130 millimeters in size. And this is on a plate. They attach to the plates. I thought I had another one that didn't come through. Anyway, and this is the third most abundant animal in Nisqually is the dock shrimp. And the size on these, 3, 2, 25, 30 millimeters. Amazing coloration. We got some amazing photographs of these guys. And I think there's a fishery for these. Pandalus platycerus is the spot prawn, which is the bigger one. But there's also a more open season for this dock shrimp too. This is a, I don't know if this is from the Nisqually or not, but this is a sort of a semi-typical plate when you bring it up. They're just loaded with encrusted animals, barnacles and bryozoans and all sorts of crazy things. Really lovely to scrape on your hand. And this is an up-close picture. So part of the project was you do an inventory of all the bigger things, quote unquote bigger, and then you scrape all these plates and put them in alcohol for later DNA analysis to identify what all of these critters are on the plate. And of course, I ran out of money and it's still in alcohol. But that's just an aspect of it. I think it's really going to be useful. And this is another plate. Oh, that's actually, so the label on the bottom is Nisqually 2018. In Nisqually, we saw 4 species of fish. The squares on here are 1 centimeter. So this is a coxcomb, Anoplarchus. And you can see it's 1, 2, 3, 4, 5, 6, 7, 8, 9 or 10 centimeters long. Really cool animal. I don't think I have a face close up. We saw a couple sculpins. This is the scaly head sculpin we saw. And then we saw a gunnel. I think we saw 4 species, maybe 6 animals total. And there was only 1 species of crab, the black claw crab. Again, these are small species of crab. This one's, as an adult, I have from Greg Jensen's book. They're 24 to 40 millimeters carapace size as adults. And I'm pointing this out because I'll show you some crab diversity at other parts of Puget Sound. And then we have, I forgot the number, 29 species of polychaetes that Leslie identified for us. Most of them, like the one on the left here, are the Arantia, the ones that run around free, versus living in a tube. This is a piece of one, piece of different ones. When we brought these up, I went, oh, who's going to identify these? But we sorted them. This was common. This was a common one. I don't remember the name of it. And then we saw a brittle star. This one's missing a leg. And that's unusual. We didn't find many of those. And we got a sea urchin as well. Again, that little red line is 10 millimeters. I'll pause here, take a breath. Eventually, maybe before you all put these into the water, if you do that, I'll have a publication with a list of all the species we saw, how many we saw, and probably some will have photographs too. So you'll have a whole inventory to start with as a baseline. And then we saw the flatworms that were really crazy. And that's as big as you could hold it in your hand, in your palm of your hand, maybe. And one tunicate sea squirt. And then this is one of the most common snails we saw. And we got as far as saying it's Amphissa. And unidentified clam. We saw pink scallops throughout Puget Sound. Again, they're pretty small when we captured them. And a limpet. I don't know what's coming up next. More limpet. Okay. This is a comparison between Shannon. Can you see the little block on the right? The Shannon, the Manchester, Nisqually, the Nia Bay, the block. Okay. So Nisqually is the green. And to read this, so how many polychaetes did we see at Nisqually? We saw 9. So that's what this bar is here, 9 polychaete species. But the number of polychaetes in Nia Bay, the purple next to it on its right, was up to 20. And actually those are the 2 areas that had the most polychaete worms. That's kind of interesting. Other things, I'll go back to crabs because they're kind of interesting. At Nia Bay in purple, there were 9 species of crabs. Nisqually, 1 species was caught, 2 Manchester in the red, and in the blue Shannon Point had 6. And I'll show you some of those species. They're really, really cool and very small. And anything else that jumps out at you? And we saw the echinoderms, the 2 species of echinoderms here, and they were absent at 2 other places. I wanted to point that out. Most abundant species everywhere, every time, was the Eulid shrimp. Although again, Manchester had almost 400 in their 3 arms, and Nisqually had 250. The others had over 100, but outstandingly abundant in South Puget Sound for whatever reason. And jingle shells, again, were abundant everywhere. And finally, I just want to point out that Shannon Point on 1 arms, we had over 140 flat top crabs. Little boogers. It was crazy. They were just crawling all over the thing. And then again, lots of Eulid shrimp and lots of the Amphissa snail. And all these crabs. Pacific lyre, graceful kelp, black claw, really fascinating little critters. Side shot of one of these. This was supposed to be a video I was going to show you. Forget it. That's not going to work. But they just get just loaded with encrusting animals and plants. And here's a side shot. So top left, I have the adult size of all these on here, 19 millimeters. That's crazy. There's reminder, 25 millimeters per inch. So these are little guys. Top left is the flat top, really ferocious looking. It had this little pad right here that helps you identify them. They're just burgundy with this pad on the claw. Sharp nose, we didn't see many of those. Kelp crab on the bottom, you'll see those. I see those swimming in Puget Sound a lot, just in eelgrass. And on the bottom right, again, the black claw that we saw in Nisqually. And that's the flat top. And again, this is an encrusted, this is an anemone all smooshed in from the Manchester spot. And that's it. So final word is, yeah, I promise there'll be a NOAA technical report that'll have all these data, at least an Excel spreadsheet. What's bogging me down is the task of trying to put these photos up into the cloud somehow in a free space. These photos of the plates can be useful to somebody. I don't know who. So I'm trying to publish those.